Hello everyone, myself Gopika Nath. Today we will be discussing about the various approaches for drug targeting, and we will be focusing on the physical and chemical approaches of targeting. So moving on to the contents, which include the introduction and targeted drug delivery system, physical approach of targeting, factors affecting physical targeting, and also the chemical approach of targeting, and the various references. Moving on to the introduction, as we already know, conventional drug delivery system, which often suffer for significant limitations such as non-specific distribution, reduced therapeutic effectiveness, and increased systemic toxicity. And so, these drawbacks are especially problematic for drugs with low therapeutic indices, high doses, and poor stability in biological environment. So, in this context, the targeted drug delivery system have emerged as a transformative approach and also which aim to direct the therapeutic agent specifically to diseased site while minimizing the exposure to the healthy tissues and also by improving the drug localization and controlling the release kinetics, the targeted delivery system enhance therapeutic efficacy, reduce side effects, and also improve patient compliance. And also among the many strategies available, physical and chemical approaches to drug targeting play a pivotal role, and also physical targeting relies on the exploitation of physiological or externally applied stimuli like temperature, magnetic fields, and ultrasound. And also, whereas the chemical targeting involves molecular modifications such as products or stimuli sensitive linkages to achieve the site specific drug activation. So, as we already know, the targeted drug delivery system is a crucial due to the limitations of conventional drug delivery system in terms of pharmacodynamic, pharmacokinetic, and pharmaceutical and pharmacotherapeutic performances. And also, the traditional methods often fail to deliver the drugs effectively to the desired site, leading to reduced therapeutic outcomes and increased side effects. So, this TDDS addresses these issues by directing the drugs precisely to the targeted site and also enhancing the therapeutic efficacy while minimizing the toxicity, especially for drugs with a narrow therapeutic indexes. And also the conventional routes like parenteral delivery is invasive, oral administration is unsuitable for protein or peptide based drugs, and also the topical applications are limited due to its local effects. And also moreover that TDDS allows drug to reach their site of action at an optimal dose and rate, reducing the systemic side effects. And also, additional advantages include simpler administration, reduced drug quantity, and also a significant increase in drug concentration at the targeted site without harming the non-target tissues. Overall, the TDDS improves efficacy, modulates the pharmacokinetics, controls biodistribution, enhances the specificity, lowers toxicity, reduces dosage, and also improve patient compliance. So, first we will be discussing about the physical targeting, and this refers to drug delivery system that localize therapeutic agents to specific areas in the body based on their inherent properties such as size, composition, or other physicochemical characteristics rather than through specific interaction with biological receptors. Unlike the biological targeting, which relies on ligand receptor binding, or chemical targeting, which may involve covalent or enzymatic modification, physical targeting exploits natural physiological barriers or pathways like leaky vasculature or the specific tissue structures to passively target drugs to the desired site. So the physical stimuli for drug targeting can come from within the body, which may be endogenous, such as pH, temperature, or redox potential, or they can be applied from outside, like magnetic fields or ultrasounds, etc. For such targeting, drugs are usually formulated using particulate drug delivery system like nanoparticles or microparticles, and these systems help in site-specific drug release because of where they physically accumulate in the body. So at the site where the drug system collects, the concentration of the drug becomes higher, which ensures the better action in that location. As a result, the amount of drug reaching other body parts will become much lower, which helps in reducing side effects. And this physical approach to targeting mainly relies on how these particles naturally behave in the body, especially when administered intravenously or orally. So, we'll be discussing the various factors affecting the physical targeting in drug delivery. First one is the molecular weight and particle size. The molecular size and weight of a drug carrier directly influence how it is distributed, retained, or cleared in the body. Small particles, which is about 30 kilodalton are usually filtered out quickly by the kidneys through renal excretion. To avoid this, carriers should ideally have a size above this threshold. And also, however, if the particle is too large, that is above 200 nanometer, it cannot easily pass through the vascular endothelium into the tissues, which is also a problem. So, in contrast, tumor tissues and organs like a liver, spleen, bone marrow have fenestrated vasculature, allowing even larger particles up to 200 nanometer to penetrate. This is the basis of the enhanced permeability and retention effect, which is also known as the EPR effect, where nanocarriers are preferentially accumulated in tumor tissue due to leaky blood vessels and impaired lymphatic drainage, enhances the passive targeting. Next one is the surface hydrophobicity and interaction with the MPS. Which is a mononuclear phagocyte system. Particles with a hydrophobic surface are rapidly recognized by the plasma proteins, which is known as the opsonins, which tag them for removal by the mononuclear phagocytic system, a part of immune system, which are made up of cells like macrophages in the liver, spleen, and lymph nodes. And also, this results in quick clearance of the carrier before it can reach the target site. And to prevent this, hydrophilic coatings, especially polyethylene glycol, are used to mask the particle surface, a process known as pigylation. And this stealth property helps avoid the opsonization and prolongs circulation time, improving chances of reaching the diseased tissue. Next factor influencing the physical targeting is the surface charge or zeta potential. The electrical charge on the carrier surface also affect the biodistribution and clearance. So neutral or slightly negative particles typically have longer circulation times, whereas highly positive charges may lead to strong interactions with negatively charged serum proteins and cell membranes, causing faster clearance and potential toxicity. However, cationic carriers are often more efficient in cellular uptake due to the electrostatic attraction to the negatively charged cell surfaces, making it a balance between stability and the efficacy. And the next one is membrane destabilization ability. For intracellular drug delivery, the carrier must escape the endosomes and release the drug into the cytoplasm. Certain delivery systems are designed to be pH sensitive, fusogenic, or include the cell penetrating peptides or viral sequences that destabilize the membranes in response to cell internal conditions. And this is especially important in gene therapy and protein delivery, where the cargo must reach the cytoplasm or nucleus to function. So, we'll be next discussing the various physical approaches for drug targeting. The first one is temperature sensitive drug delivery system. This works by using heat to control where and when a drug is released. Many diseased tissues like tumors have slightly higher temperature than the normal tissues, a condition which is known as hypothermia. Also, heat can be applied externally, like the magnetic fields, to rise the temperature in a specific body area or trigger the drug release. So Mayer et al. Showed that when tumor area was heated to up to 42 degrees Celsius, liposomes and other drug carriers accumulated more in the tumor. This was tested in a model of human ovarian cancer in mice. And also scientists use temperature-sensitive polymers to make this system. These polymers have a special property called LCST, which is a lower critical solution temperature. Below this temperature, the polymer dissolves in water, and above it, the polymer becomes insoluble and changes the structure. And this helps in releasing the drug from the delivery system. And also, Nakayama et al. Developed missiles using the block polymer of polynipam mixed with biodegradable materials like PLA and or PCA. And these missiles released doxorubicin quickly when the temperature was raised, and the materials used were biodegradable and making them more safe in delivery. And the next one is the magnetic sensitive drug delivery system. The magnetic sensitive systems were first introduced by Wider et al. In the year of 1979, and the concept includes attaching the drug molecules to magnetic particles using an external magnetic field to direct them to the target site. And also, these systems commonly use super paramagnetic iron oxide nanoparticles, which is pions, such as magnetic or magmite, which are very small, which is about 4 to 10 nanometer and exhibit strong magnetic responsiveness. For example, Gang et al. Demonstrated the use of magnetic polycaprolactone nanoparticles carrying gemcitabine for treating pancreatic cancer in mice, while Alexia et al. Successfully targeted mitochondrial loaded spions to squamous cells carcinoma in rabbits using magnets. Next is the ultrasound sensitive drug delivery system. These systems work by allowing the nanocarrier to accumulate in the target site where the external ultrasound is applied to make the tissue more permeable. And this causes nanocarriers to break apart and release the drug or genetic material they carry. And for instance, polymeric missiles loaded with anti-cancer drug doxorubicin have shown drug release when exposed to ultrasound. And similarly, Tikonhoy Liang et al. Used ultrasound to release thrombolytic enzymes like plasminogen activator from spatial liposomes at blood clot site. And in this case, the drug not only targeted by the clot by binding to fibrin, but also served as a contrast agent, making the system multifunctional with imaging, targeting, and therapeutic capabilities. Next one is the shape, size, and surface touch manipulation. So the mechanism involved red carriers like nanoparticles, liposomes, or microparticles are engineered with specific sizes, typically which is around 10 to 200 nanoparticles per nanoparticles, and the surface starts to exploit the enhanced permeability and retention effect in tumors or inflamed tissues where the blood vessels are leaky. And examples include liposomes and polymeric nanoparticles accumulate in tumor due to EPR, passively targeting the drug to cancer cells. And next is the light activated release, where the carriers incorporate photosensitive materials or polymers that release drug upon illumination with UV, visible or infrared light. Light enables high spatial and temporal control. Example include nanoparticles with poly to nitrobenzyl methacrylate degrade upon UV exposure to release the drugs, and near infrared light triggered systems penetrate deeper tissue for localized therapy. Next is the chemical targeting for drug delivery. Uh, the chemical approach to drug delivery focus on designing system that ensures safe and targeted delivery of drugs to specific sites in the body. And this method involves modifying the drug molecule so that it becomes active only after undergoing a predictable enzymatic reaction at the target site. And additionally, certain chemical groups may be added to the group in order to deactivate or detoxify it after it has produced its intended product or effect. And in the system, the active drug is chemically linked to one or more carrier molecules, and if needed, protective groups forms a site-specific delivery system that ensures precise and controlled action. So the first is the Prodrug concept, which we already have studied. Uh, where the chemical targeting involves using prodrugs, inactive forms of the drug that become active only after undergoing specific chemical or enzymatic reactions at the disease site. And this selective activation minimizes the side effects or on the healthy tissues present in the body. And also, there are two advanced strategies using this concept. The first one is the antibody directed enzyme product therapy, which is also known as ADAPT, and also gene directed enzyme product therapy, which is the GDAPT. So the first approach, which is the antibody-directed enzyme product therapy, uses an antibody attached to a special enzyme that specifically targets the disease cell. And after this antibody enzyme complex reaches the target site, a prodrug is given, and the enzyme activates the prodrug into its toxic form only at the disease location, destroying the harmful cell while leaving the healthy one unaffected. So the gene-directed enzyme product therapy works by inserting the drug into the tumor cells that produce specific enzymes, and these enzymes then activate the product only within the tumor. And also this targeted activation, particularly effective in low oxygen or the hypoxic tumor areas, helps in the drug act precisely at the disease site, minimizing harm to healthy tissues. And also, for example, in GDAP, the gene for the enzyme HSP thymidine kinase is inserted into cancer cell. When ganciclovir, a nucleoside analog, is administered, it is converted into monophosphate formed by HSP-TK. And also, this is further phosphorylated into triphosphate, which disrupts the DNA synthesis and triggers apoptosis. And this approach is known as a suicide gene therapy, and HSP-TK is referred to as a suicide gene here. And additionally, this method causes a bystander effect where even nearby non-modified tumor cells undergo apoptosis, enhancing the overall tumor destruction. Since it directs action to specific cell, this approach also qualifies as a form of active and combination targeting. And this is the image which shows the concept of chemical targeting to cancer cell, and the product here is not activated in the normal cells and due to anaerobic conditions. Next is the enzyme activated products. The enzyme activated products are specially designed to overcome active only in the presence of certain enzymes that are found in high amounts in tumor tissues, and this helps ensure that the direct targets cancer cell while sparing the healthy ones. And also, for example, gemcitabine used to treat pancreatic cancer is an inactive product, and that is converted to its active form by the enzyme deoxycytidine kinase, which is abundant in the pancreatic tumor cells. And once activated, the gemcitabine kills the cancer cell by interfering with their DNA synthesis. Next is the pH-sensitive drug delivery system. The pH-sensitive systems are one of the most widely studied smart delivery approaches. They take advantage of the fact that diseased tissues such as inflamed areas, infects or tumors have a lower pH than normal healthy tissues. And for example, the normal tissue which is healthy may have a pH which is about 7.4, while the tumor tissues and the sites of inflammation have a pH around 6.5, which is lower than the healthy tissues. And this acidic environment is mainly due to low oxygen levels in tumors, which leads to the production of lactic acid. So this property is used to design a drug or gene delivery system that release contents only in the acidic condition. This helps in targeting the drug specifically to the disease site, reducing the side effects in healthy tissues. And to achieve this system, drugs can be attached to pH-sensitive polymers using acid-sensitive spaces, and these spaces used to break down when exposed to acidic pH, releasing the drug either in the extracellular space near the tumors or inside the acidic cell compartments like endosomes or lysosomes. So one example is Camara et al., who developed pH-sensitive polymer carrier using polyvinyl pyrrolidine dimethyl malic anhydride linked with doxorubicin. The system released doxorubicin in response to the acidic tumor environment, and which showed better accumulation at the tumor site. And another group used a special polymer called polybeta amino ester that dissolves at only pH below 6.5. They made nanoparticle with this polymer and filled them with paclitaxel, a drug which is used for cancer. This nanoparticle showed more drug inside the cancer cells and tumors, and better cancer cell killing in lab tests, and a smaller tumor size in animals compared to the regular paclitaxel solution. So next is the redox sensitive system. This take advantage of the difference in redox potential between the inside and outside of the cells, and the extracellular space is more oxidizing while the intracellular space is more reducing, especially because of the high levels of glutathione inside the cells. So these systems often use disulfide bonds to keep the nanocarrier intact. When the carrier enters a cell, the high concentration of glutathione breaks this disulfide bonds into thiol groups, and this causes the carrier to fall apart and release the drug or the gene which is delivered. So, for example, Cavallaro et al. Used a positively charged polymer with thiol groups. The polymer carried DNA through electrostatic attraction and formed a network via disulfide bonds. So, once inside the cell, the disulfide bonds broke and the network got disintegrated and the DNA was released at the target site. And next one is the Kirpotin et al. Designed redox sensitive liposomes that are coated with PEG, which is polyethylene glycol, linked through a disulfide bond in the reducing environment. Inside the body, the PEG coating was detached, allowing the liposomes to release the drugs while still showing long circulation in the bloodstream. And now we are moving on to the summary and conclusion. Both physical and chemical targeting strategies offer promising solutions for overcoming the limitations of conventional targeted drug delivery systems. And the physical approaches utilizes physiological characteristics or external stimuli to guide the carriers to specific locations, enabling passive or externally triggered delivery, as we have already discussed, like the temperature, the usage of temperature or magnetic field or ultrasounds, etc. And the chemical targeting, on the other hand, relies on molecular modifications that ensure that drugs become active only at the disease site, often using enzymatic redox or pH-sensitive triggers, so which reduces the effect on the healthy cells. So, when appropriately designed and applied, these methods not only improve the localization and efficacy of therapeutics, but also reduce the systemic toxicity and enhance the patient outcome and improve the patient compliance. As research continues to evolve, the integration of both approaches, along with advances in nanotechnology and biotechnology, is expected to revolutionize precise medicine and pave the way for more personalized and efficient drug delivery system. So these are the various references. And Thank you.